Hello everyone, my name is Leonard. Thanks Paul for the generous overview of our association. Yes, I'll introduce our speaker for tonight a bit more briefly than Paul just introduced our association. Professor Dr. Paul Cumming. He is an author, a neuroscientist, and a professor in the department of, let me get this straight, there's this one German word. It's... Oh, thanks. Okay. Oh, okay. I'm relieved. Nuclear medicine. Nuclear, that's the word. Exactly. And he researches on molecular and neuroimaging techniques, especially positron emission tomography. You might have heard the word PET scan. That's what it stands for. And he uses this technology to study neurotransmitters. Among others, that's serotonin and dopamine, importantly. And he uses this knowledge and these technologies to also study neurodegenerative disorders and psychiatric conditions such as Alzheimer's or Parkinson's. Professor Cumming has held prestigious positions at well-known universities all around the world, amongst others, McGill University, Oslo University, Aarhus University, and Queensland University of Technology in Australia. In more recent years, Paul Cumming has also published extensively on other neurobiological fields, such as psychedelics, including MDMA, psilocybin, LSD, and DMT, and also MAO inhibitors, such as the beta-carboline harmine. In tonight's talk, Paul will give an account and discuss the neurochemistry of DMT, which is an important constituent along with the so-called beta-carbolines, harmine, harmaline, or tetrahydroharmine, that are three beta-carbolines commonly found alongside with DMT in the plant-based brew called ayahuasca, which is found in the Amazon basin. Please give a warm round of applause for Professor Dr. Paul Cumming.
Okay, am I live? Yes, good. Thank you for that introduction, Leonard, for inviting me, and thank you, Paul, for the general introduction. So the scale of this is astonishing for me. It reminds me a little bit of, as an undergraduate in chemistry in Canada, my homeland, there was a regular seminar series for the graduate students. Usually 10 people would come and listlessly wait for it to be over, but there was one person who did a graduate project on a total synthesis of cocaine, and the auditorium was completely full. So, drugs turn heads somehow, but also I'm sure it hasn't escaped your notice that this kind of interest that you represent in this topic of psychedelics is a legitimate and important topic for research in basic sciences and from the point of view of clinical applications in a variety of conditions, is such a recent phenomenon, and you only have to do PubMed searches to see the biphasic course of publications in this general domain, which kind of peaked in the 1960s, as you might imagine, and then due to, frankly, political decisions, it was out of reach for nearly decades until this enormous growth of development in the past decade. And we're playing catch-up because there are basic issues that still have to be sorted out, very simple things about the chemistry and neurochemistry of psychedelic compounds, let alone the applications in clinical investigations. And so I'm going to talk a little bit about my personal involvement in trying to catch up, that is to obtain a basic understanding of the biochemistry and neurochemistry of, in particular, DMT. And so DMT, of course, stands for N,N-dimethyltryptamine, which is a constituent of ayahuasca, probably the key constituent. And as I'm sure you all appreciate, as is well known, the DMT, when taken orally, although it's a potent psychedelic substance, when administered by insufflation or intravenous injection, short-lived but potent. When taken orally, DMT is practically inactive. You can eat it literally by the spoonful, I think, if you wanted to, and it would do nothing because it is subject to extremely extensive first-pass metabolism through the gut, and the principal culprit for that is monoamine oxidase, an enzyme that is ubiquitous in many organ systems and in the brain, particularly the monoamine oxidase type A. And so somehow or another, nobody knows how, the indigenous peoples of various parts of Amazonia, Peruvian and Brazilian Amazon, I guess, discovered that a decoction of two plants had the remarkable property of producing these psychedelic effects. One of the plants containing the active constituent DMT, and the other containing a mixture of various alkaloids, not, as far as we know, intrinsically psychoactive, but acting as potent inhibitors of monoamine oxidase type A, and that is the beta-carbolines that Leonard mentioned. And taken in conjunction, of course, the blockade of MAO by one compound massively potentiates the absorption and plasma half-life of the active constituent DMT. Just to put it aside, it is not completely certain that the harmine and beta-carboline alkaloids that principally act as monoamine oxidase inhibitors absolutely do not possess other psychoactive effects. But in the main, we consider them to be acting as MAO inhibitors. And when taken in conjunction, producing the, potentiating the psychedelic effect of DMT. So my interest in this was drawn actually decades ago in the context of work that was done from the famous and pioneering laboratory for PET research at Brookhaven National Laboratories. And this is a famous figure dating from, I think, 1990 or so. You know, it's that old, where Joanna Fowler, I think her name was, had been working on a ligand that is a radiopharmaceutical that is radioactively labeled so that it can be seen by positron emission tomography, and was, by its virtue of its structure, targeting specifically the enzyme monoamine oxidase, here in particular the B type. And that is deprenyl, labeled with carbon 11. More about that later. But when injected intravenously, deprenyl, labeled with carbon 11, distributes throughout the body, passing through the circulation and depositing in various organs and sticking in various organs in measure of the local abundance of the enzyme, target enzyme monoamine oxidase. And you see on the left-hand side the sort of image obtained by passing the patient through the scanner in a moving bed to acquire an image capturing the radioactivity through the entire body, that there's substantial trapping of the ligand in brain, where we know there's lots of MAO because MAO is involved in neurotransmitter metabolism, but it's also highly abundant in the heart, where it's also probably involved in the sympathetic nervous system regulation, and the liver and the kidneys, which have high concentrations of the enzyme. However, they incidentally noticed in one of these, you know, the saying, Pasteur said that Lazare favorise l'esprit préparé. They somehow noticed that a particular participant who was a heavy smoker had practically no uptake of the radiopharmaceutical in the brain. And they repeated this in a series of smokers and determined that, yes, indeed, people who smoke have a substantial inhibition of monoamine oxidase, actually both types, A and B. And it occurred to me, and to others, I suppose, at the time, that this might contribute to the psychopharmacology of tobacco smoking, which is usually attributed to the effects of nicotine on the nervous system. But if the nicotine is acting in conjunction with monoamine oxidase inhibition, it's acting upon a nervous system that is primed for perhaps more potent activation of noradrenergic, serotonergic, and dopaminergic transmission. And so I put it this way, yeah, that the Brookhaven work inspired my interest in ayahuasca dating back all this time. Does the MAO inhibition from tobacco smoke alter dopamine or serotonin metabolism in brain of smokers, perhaps potentiating or acting synergistically with the main effect of nicotine? Is smoking ayahuasca light, in other words? I think so. I did some experiments in pigs trying to establish that, but it proved an elusive subject. But it's hard to imagine, I think, that having such a substantial blockade of monoamine oxidase in your brain would not have some kind of inherent psychopharmacological effect. So my student, my shared student, Clemens, who may be known to some of you, recently, this came out I think a week ago, did a massive review summarizing what is known about DMT metabolism and its effects on the brain and its interaction with MAO inhibitors. And oh, there it is, yeah. And I mean, I think it's a monumental work that he and Helena put together. I forget how many pages it is, but it's really comprehensive. And so I take the liberty also of using one of his fine figures to introduce you to the structural aspects of this class of psychedelics. And here we see the star of tonight show, which is DMT, and it contains, by comparison with other psychoactive compounds and the natural neurotransmitter here, serotonin, a considerable degree of similarity with other tryptamines, many of which are psychedelic. Even LSD has a rigid orientation of tryptamine embedded in its structure. And obviously, or evidently, something about that indole structure lends itself to interacting with serotonin receptors in the brain and mimicking or exaggerating the effects of natural serotonin seems to be part of the nature of DMT and related psychedelics. And we have to make the distinction between the other class of classical psychedelics, which is the phenylethylamines, like 2CB and mescaline, which have a different structure but similar targets. The other player in ayahuasca, as far as we know, is the beta-carboline alkaloids, which, if you look closely, they are also indoleamines that are cyclized. Probably, and just by the way, the MAO, part of the MAO inhibition in brain of smokers is derived from the combustion product or pyrolysis product of tryptophan in the combustion of tobacco. So it's actually not present in the tobacco until you burn it, and then you get this formation of cyclized indole compounds that are belonging to the category of beta-carboline MAO inhibitors. So there are a number of other structural analogs that in varying measure exert this monoamine oxidase inhibition effect. And another one of his Clemens diagrams summarizes the various pathways that are possible. You see that the Banisteriopsis plant, for example, from South America contains not just harmine, shown here, but a number of other beta-carboline alkaloids, which are also monoamine oxidase inhibitors and probably exert other effects as well. This is all very vague because everyone has assumed that the only thing that beta-carbolines do is block MAO, but it's probably not limited to that. There's an entourage effect going on with, if I may say, a witch's brew, such as ayahuasca, which contains so many psychoactive compounds. But the main effect, the principal component that is responsible for MAO inhibition is harmine, shown here, and it's acting upon this reaction whereby the psychedelic substance DMT would otherwise be broken down by monoamine oxidase type A to yield this so-called acidic metabolite, hydroxyindole acetic acid, which is a carboxylic acid, as you can see here, no longer psychoactive, and it's rapidly eliminated via the kidney. So that's the basis of the simple reductionist view of the psychopharmacology of ayahuasca. Serotonin 5-HT2A receptors, as you know, are thought to be the common site of action of DMT, psilocybin, and LSD, but all of these drugs have different overlapping and complex properties, activating or inactivating a number of receptor types. So you cannot say a priori that the only unique action of classical psychedelics is through 5-HT2A receptors, but probably it's the composite, especially I think with LSD, which binds to so many different targets. So Focusing, though, for the time being on the 2A receptors, which are clearly linked to the psychedelic effects. Where are they found in brain? Well, positron emission tomography, or PET, came to the aid of answering this question, where you can label a particular molecule, such as a pharmaceutical that has high affinity for the target in question, here 5-HT2A receptors. And if the person is positioned in the scanner, here the very first PET scanner, I think it was produced in the 1960s, you see that there's a ring of detectors, and the decay event takes place someplace in your head where the photons are released. And so you detect with the detector ring the emission of high energy photons from this positron electron annihilation. So the basic principle of PET is antimatter. The mode of decay of the radionuclides used in PET imaging, such as carbon-11, is by the emission of a positron into the medium of the brain, which is an antimatter electron. And it passes some distance, like in a pachinko game, bouncing here and there, and eventually it encounters a native electron at just the right angle and kinetics that they meet and, so to say, embrace in what is called a positronium atom. It's like an actual atom of an antimatter and normal matter electron, but it's for some reason related to the nuclear physics, it's not stable, but rapidly they coalesce and annihilate each other. And they go bang, basically, and all of the energy, E equals mc squared, bound in that electron-positron pair, is emitted as two high-energy photons, or gamma rays, at pretty much 180 degrees from each other. So if you have a ring of detectors, if two detectors go off in the same nanosecond, that counts as an event, a decay event. And by counting millions and millions of such events, it's possible to reconstruct an image, a source map, showing where the radioactivity was at the time of decay. And using this method, my colleague in Vienna, Rupert Lanzenberger, and his group labeled a molecule called altancerin, which is a serotonin antagonist, with fluorine 18, the positron-emitting isotope, and here produced this map showing the distribution in human brain, and you can see that it's a cortical binding site. It's found in the frontal cortex, a little bit less in the associative cortex here, but very abundant in the visual cortex back here, and hardly present or nearly absent in the subcortical structures. So at least the effects of psychedelics on serotonin 2A receptors is a cortical phenomenon, because that's where the receptors are found. Just restating that the serotonin 2A receptors are mostly found in the cerebral cortex. There's at least 8 different serotonin receptors, and they have different distributions. But here we're talking about 2A. So when I was in Aarhus, Denmark, I was interested in this topic, and it occurred to me that the beta-carboline harmine is not just an interesting molecular entity, but it's also amenable to labeling with carbon-11, the positron-emitting nuclide, in the laboratory, so that you can then use it as a probe to see where MAO is present in the brain. And a friendly radiochemist there was able to develop a radiochemical synthesis from a carbon-11 produced in the cyclotron in the morning of use, because with a 20-minute half-life, after which with each 20 minutes the carbon-11 that you start with decays by 50%, you can imagine that this is not for sleepy chemists. They have to make it and synthesize it in the molecule and purify it in a state ready for human administration to humans, all in a matter of an hour. And it so happens that this particular reaction is facile and efficient. And so they were able to prepare it for my pig studies. And other people in Toronto used the same ligand, harmine, the very same harmine as in ayahuasca, as a tracer in a variety of human studies, especially in depression. So here's what you get. These are parametric maps, and it shows, in effect, the distribution of the harmine in the brain of not one pig, but 3 pigs, averaged, because pig brains, like human brains, are so similar that you can basically stack them one on top of another and average them. And here in the lower part of the figure, a blocking study where the same pigs were scanned again, but after being pre-treated with a very potent blocker of MAO. And you see it left practically nothing. But in the unblocked condition, you can see where the MAO is present in the brain. And it's throughout the cerebral cortex. It's also highly abundant, and this is the thalamus, which is the gateway part of the brain and a site of action of psychedelics, certainly. In the hypothalamus, in the basal ganglia, in other words, in serotonin-rich areas. MAOA is associated with, not perfectly, but it has a considerable overlap with the distribution of serotonin in the brain. Why? Because it's the enzyme that metabolizes serotonin. And that points to its key function, not just in psychedelic action and DMT metabolism, but that of natural serotonin. So any DMT molecule that has survived first-pass metabolism in the gut and passes into the brain is not completely off the hook, because it's also being exposed to all that abundant MAO, very high activity in the living brain. So even once entering the brain, a DMT molecule is rapidly broken down unless you block the MAO with a brain-penetrating pharmaceutical. So we mustn't neglect the importance of cerebral metabolism of DMT, whereas most of the emphasis has been placed on the limiting effect of gut metabolism on the absorption of DMT. It's also being metabolized by MAO in the brain as well. You know this toad, right? I think it's threatened with extinction because people are chasing it. And it's not for its cute smile, but for the DMT-like molecule, bufotenin, in its skin, in its the glands in its back, which obviously has a considerable structural overlap with serotonin and with tryptamine and DMT itself. And you probably know, I don't know much about DMT, about this compound, but it has a reputation of being even more potent than DMT itself. And anything with a hydroxy group, any molecule with a hydroxyl group on it, you can consider as being available for methylation. And for a radiopharmaceutical chemist, that means preparing a PET tracer with carbon-11 methyl, because using this reagent here and others, it's possible to react very efficiently to produce the, in this case, meo-DMT, 5-methyl-methoxy-DMT, which I'm sure you've heard of, but it's labeled with carbon-11. And so when I was in Denmark also, this was, 20 years ago, I guess, it occurred to us that this should be an interesting molecule to test in our pig model to see where it goes and whether it sticks in the brain. And in brief, we obtained dynamic PET recordings, dynamic meaning the time series starting at the point of an intravenous injection of the meo-DMT for 60 minutes, and took blood samples so that we could analyze the metabolites, and then in effect calculated binding maps of the distribution volume of the tracer in brain, which was supposed to show where it's sticking in the brain, in the way that some of the other tracers have worked. And then do certain pharmacological challenges to see if you can kick it off its receptors. So my hypothesis was, and it was probably a weak hypothesis really, but the idea was that meo-DMT would reveal the distribution of serotonin receptors in the brain of the pig, because that's where it's acting as a psychedelic. Of course, these pigs were anesthetized, so they missed the fun part. But and of course, we don't know for sure that anesthesia, to get them to sit still for the scan, doesn't interfere with metabolism and kinetics. That could also be. So you remember what the altancerin binding looked like in the human cortex, in the human studies? I showed you a figure, and it was pretty much exclusively found in the cerebral cortex and practically absent from the subcortical structures like the striatum. And what we found unequivocally, this is the mean of 3 determinations in separate pigs, that it really didn't have any binding in the cortex at all, but was binding in the striatum. Specifically, you know the striatum, that's the dopamine-rich part of the basal ganglia, which is affected in Parkinson's disease. And the striatum doesn't contain very many serotonin 2A receptors, so this was very curious. And we tried the classic displacement study where you block with a non-radioactive pharmaceutical to occupy the binding sites, and therefore if it's targeting the same site, the catancerin treatment, which is a serotonin antagonist, should have knocked off the binding. But you can see by looking it did nothing, it didn't touch it. So DMT was a failed tracer. And it was never, I was never able to explain this phenomenon because it's clearly binding, sticking somewhere in the ventral striatum, which is the reward locus of the nucleus accumbens, if you want to know. And could that actually be indication of a dopaminergic action of DMT? And this is autoradiography, which is like the poor person's PET, where it's done in thin brain sections incubated with another kind of radioactive ligand. And here they used a ligand specifically binding to D3 receptors in the rat, and you can see that the D3 dopamine receptor is only abundant in the ventral striatum. So I ran out of funds for that study, but my guess is that a D3 blocker would have displaced this binding. But there's more sadness in that I never managed to get this published because I think I tried 5 or 6 times. But it was always rejected because the result was negative, and nobody likes a negative result unless it's a COVID test. And I guess I should actually resubmit it finally after all this time, because now it might pass. Unfortunately, I'm not in a position to do any new further experiments with this tracer. But it's interesting. So now I'm going to shift. I do have to mind the time. Oh, I'm okay. One of the most popular targets for molecular imaging by positron emission tomography is glucose metabolism. So far, I've talked about receptor ligands that are binding to some neuroreceptor, especially serotonin receptors. But by far and away, the most common use of PET is for diagnostic purposes, because most tumors, many tumor types, are avid for glucose. And this particular tracer, labeled with fluorine 18, another positron-emitting nuclide, mimics natural dopamine and enters living tissue and is trapped in measure of the local rate of glucose consumption. And so in this unfortunate person, you can see that there's a FDG, fluorodeoxyglucose, avid mass in the pulmonary artery here. That's a tumor. But incidentally, you see, they weren't interested in the brain and they cut it off in the scanner. You can see that the brain is even more hungry for glucose than tumor is, and that's characteristic of healthy brain tissue. I did a it's well known that the brain is burning about, how much? 300 watts. Something like 20% of your energy budget of what you eat goes to just keep the brain, even when you're asleep. And this is a strange thing, by the way, about the brain, because it doesn't need to be active to be desperately hungry for glucose, because the upkeep of neurons is inherently expensive. And so the brain has a prodigious consumption of glucose. And that shows up in this PET scan by showing how dark the human brain is. And this led, Franz Vollenweider did this pioneering work more than 25 years ago, when nobody else was doing it, basically. And he used fluorodeoxyglucose, FDG, to answer this very simple question: what does psilocybin do to brain metabolism? Does it increase the rate of energy consumption in the brain by stimulating it in some way, or does it decrease it, or does it have regional effects? And so it was, that was the hypothesis. They gave psilocybin, I forget what dose, 30 milligrams to healthy volunteers, and scanned them before and after. At baseline, and of course they're awake because they're healthy volunteers, and during the psilocybin experience. And this is nothing to do with psilocybin, it's just to show you that FDG PET gives maps where you can see the regional pattern of and magnitude of glucose consumption, high in some areas, low in others. how does psilocybin alter that? Well, what he found, and nobody sees any reason to doubt him because they saw what they saw, was that there was a 15 to 20% increase in glucose consumption practically everywhere. These are all different brain regions in both hemispheres. In other words, psilocybin greatly stimulated brain metabolism globally. And this was unexpected. Nobody has replicated it really since all that time. We're getting to that. So could psilocybin really increase brain metabolism, glucose metabolism so much? To kind of get generalized from that result with psilocybin, that is my graduate student, Clemens Eggers, and colleague in Denmark, Michael Palner, who's a rising PET researcher in psychedelics, did a little study in rats to test the effect of DMT harmine mixture on cerebral metabolism in rats, anesthetized rats. And this is from the figure from his paper on the subject. And you see that in the normal rat, at baseline, the vehicle-treated rat, these are different levels, horizontal slices we call them. This one's slightly more ventral than this one. So you can see there's high glucose uptake in the frontal cortex and the cingulate cortex, and lesser uptake in some other structures, high uptake in some thalamic structures. This is all well known. So did these treatments, harmine, DMT alone, or harmine and DMT, did they alter glucose metabolism in this group of rats? We only had 6 per group, which actually is not enough because the results are so variable. But just looking at these average images, each of these images is for the vehicle treatment group, and the harmine, DMT, and harmine plus DMT groups represents the mean of 6 rats that are stacked up and averaged in the common stereotaxic space, as we call it. And really, to visual evaluation, doesn't look like much is going on. There wasn't a 20% increase, at least. However, on close examination, here in the vicinity of the thalamus, you see it's hot here, and then it's cooler under the different drug conditions. Something might be going on. We have to go further and increase our group size to nail that down. But it did look like, here are the actual numbers for the semi-quantitative uptake of the radioglucose, and it did look like there was an increase in the thalamus only with the co-joint administration of harmine and DMT. It was nowhere near significant because of the high variance, but it was in the right direction anyway. So maybe a little bit of caution, and also to myself, statistical power is very important. To draw firm conclusions, you need to have enough power. With that in mind, we're now proceeding and well underway with the main part of the study, an SNF-funded grant, to investigate the cerebral metabolic effects of harmine DMT in healthy humans, using pretty much exactly the same protocol as Franz Wollenweider did 25 years ago, with a baseline scan and a post-drug scan. But this time we're giving DMT harmine, and we're using, well, let's just say PET scanners are much, much better than they used to be. For one thing, we can capture the whole brain and the whole body from your knees to the crown of your head, the vertex, and produce maps like this using the new whole body PET scanner in Bern. And you can see what might be a familiar pattern now, that there's high uptake in the brain and in the heart, and this is the bladder because it goes into the urine. So we're going to do this ultimately in 16 volunteers. So far, I think we scanned 11, and so we're still in the data acquisition phase. But the objective is to test in an exploratory manner with sufficient subjects, n equals 16, the whole body effects of DMT harmine on glucose metabolism. In returning to the rat study, we actually measured the DMT and harmine in the brain, which is something you can't normally do, except in rats. And that was in collaboration with the forensics departments here. But the rats were given different doses of DMT and harmine, and then basically off with their heads, and brain regions dissected and preserved for measuring the content of harmine and DMT. And the objective here was simply to confirm that harmine actually does potentiate the uptake of DMT into the brain. As it did, I think basically, I won't ask you to really examine this table closely, but well, here's the DMT levels. We see traces of DMT in the vehicle-treated animals. And that's probably real because, as you have no doubt heard, DMT is actually produced in the brain, but it's rapidly broken down, so it never attains high levels in the brain. And giving DMT alone really had no effect because the DMT given, I think, subcutaneously, never got into the brain in the first place because of the MAO in the body. But giving harmine alone had little effect, but giving harmine and DMT together produced a massive influx of DMT into the brain, really 15 to 20 times what is present in the control condition. And we did various other experiments to measure the DMT metabolite, 3-indole acetic acid, 3-IAA, and show that there were sort of parallel effects. That if you give, for example, DMT alone, without giving an MAO inhibitor, there are massive amounts of 3-IAA, the DMT metabolite in brain. In other words, the DMT does get into the brain, even without MAO inhibition, but as soon as it enters the brain, it's immediately broken down by MAO in the brain, yielding this metabolite IAA. And we also measured the actual harmine concentrations and confirmed that they were enough to substantially block MAOA. So focusing on a small part of that table, this is something I would like to draw to your attention. We measured the DMT levels in brain, and under the condition of 3 milligrams per kilogram with harmine, there was 10 micromolar. Now 10 micromolar is a high concentration for neurochemistry, and when we consider that the Ki of DMT, that is the sort of affinity of DMT for serotonin receptors, is 100 times lower, 100 nanomolar, it follows that with such a high DMT concentration, it really should be sticking to the serotonin receptors, yeah? Stands to reason. And how to assess that? Well, we did it ex vivo. This was again an experiment hosted by Mikkel Palner in Odense, Denmark, and we measured the occupancy ex vivo, where we gave a radioactive serotonin ligand in control animals and in animals treated with harmine, DMT, and harmine plus DMT, and then measured the specific binding in the animals without doing a PET study, just in the tissue samples. And so here is in effect, the binding ratio for serotonin receptors in the cortex in the vehicle animals and in the harmine DMT and harmine plus DMT. Now, there was no effect. So we weren't able to measure occupancy at serotonin receptors in the living brain, even though we know that the DMT concentration here was something like 10 micromolar. So this is weird. We don't know how to interpret that yet. To summarize, the cortical binding of ketanserin, that was one of the tracers we used, labeled with tritium, was exactly the same in the harmine and DMT-treated rats as in the control rats. In other words, there was zero measurable occupancy. So how can DMT be acting at serotonin receptors if it's not binding to them? Even though the concentration of DMT in the brain was so high that it really should have been sticking. We have some explaining to do. And in an elegant study by Gitte Moos Knudsen, who's kind of the leader in psychedelic brain imaging these days in Copenhagen, they used another serotonin ligand showing this high binding in the cortex at baseline, and then administered psilocybin at some dose, 30 milligrams. And you can see, as clear as day, the psilocybin has blocked most of or half of the serotonin receptors, showing target engagement by virtue of displacing the radioligand and competing in competition for the same binding sites. You can see, is everyone comfortable with that concept of a competitive binding study? So here's the baseline, and here's post-psilocybin. And furthermore, the individual degree of occupancy, in this individual it was sort of 50%, it correlated with subjective effects. So the greater the occupancy in an individual, at the serotonin receptors, the more intense their experience. And so that makes it even more peculiar why DMT failed to evoke discernible occupancy so far. But people are working on it. We'll get it sorted out eventually. Is there some methodological reason why we couldn't discern the occupancy? Yeah, probably. And I think I don't really have time to go into that, but it might have something to do with the fact that DMT is binding to a whole lot of different receptors. The 5-HT2A may be the most important one, but I don't think we can say that that's the only site of action for DMT or other psychedelics. So now I'm going to talk very briefly about some new rat studies that I did to try to get a handle for what happens to DMT in the living brain. And we know that when you administer DMT, it gets into the brain and it obtains certain concentrations, but that in the brain it's broken down by monoamine oxidase A resident in the brain. And unless you block MAO, it kind of is a short circuit. As soon as it enters the brain, it's immediately converted to the inactive metabolite 3-IAA. So this is a typical diagram showing a generic serotonin synapse where tryptophan is the precursor for serotonin synthesis. It's stored in vesicles. It's released synaptically, transsynaptically to bind to inter alia serotonin 2 receptors, and then is taken back up again by the serotonin transporter and can be recycled. So that's the fate of natural serotonin in this pathway. And DMT, of course, closely resembles serotonin in its structure and it's in its regard to its affinity for certain transporters. In particular, there is in animal evidence that DMT is a substrate for the serotonin transporter, both in the plasma membrane and in the vesicles. So my idea was, okay, maybe DMT is behaving strangely because it's hijacking the serotonin terminals and actually replacing serotonin by pushing it out of the way. And that would be summarized here in the way to get at that. A very simple, very basic neurochemistry experiment is to try looking at the disposition of DMT in the rat brain after pharmacologically blocking those two different processes with escitalopram, that's an SSRI, a serotonin uptake blocker, an antidepressant, which disables the serotonin transporters. So any DMT in the brain would then no longer be able to get into the serotonin terminals. Or in another study, use this compound dihydrotetrabenazine, which is a form of reserpine, which blocks the uptake into the vesicles. So we could approach this pathway from either side, from the plasma membrane side or from the vesicular side. And if DMT is in fact being stored in vesicles of serotonin neurons, then both of these drugs should inhibit its retention in brain, yeah? Nice idea? I thought so. And we again did this trick, and here I dissected the brains into 7 regions and froze them and sent them to Zurich for analysis of serotonin and its metabolite HIAA in the 3 conditions, from left to right, dihydrotetrabenazine, citalopram, the SSRI, and the control animals. And you can see that serotonin concentration is highly variable between brain regions. There's practically none in this brain region and very high levels in this brain region, which is the midbrain. And that's normal, that's to be expected. And the serotonin metabolites formed by monoamine oxidase, 5-HIAA, more or less track the serotonin levels. So the brain regions with high serotonin also have high levels of the serotonin metabolite. And again, I emphasize that serotonin, when it's in a vesicle, is safe for metabolism. But when it passes through the cytosol of a serotonin neuron, it's exposed to monoamine oxidase in the neuron and therefore is broken down to the metabolite. And they generally have a kind of a steady state ratio. Now, basically, the drug treatments had the expected effects. And we don't have time to go into them in detail, but dihydrotetrabenzene, which blocked the vesicular storage, depleted serotonin in the brain by 80% and augmented the amount of metabolism because it doesn't get into the vesicle, so it spills out into the cytosol and is broken down. And it basically across the board, both drugs had the expected effects on serotonin. And again, I remind you that serotonin levels, endogenous, the actual neurotransmitter serotonin levels, differ substantially across brain regions in measure of the density of the serotonin innervation, which is very sparse here and very abundant here. And so the same animals were treated with DMT and harmine, and we measured in the same, that is to say, the colleague at forensics who has the setup for measuring these compounds, measured DMT and its monoamine oxidase metabolite. And the whole idea was I was thinking DMT should be behaving just like serotonin vis-a-vis its distribution in the brain and its sensitivity to these drug treatments. But what we see basically is that the DMT concentration is flat. It's the same everywhere in brain. So its distribution in the brain has nothing to do with the local serotonin innervation. And furthermore, the HIIAA levels, maybe with the exception of this brain region, are also flat. So DMT simply goes into the brain, distributes equally in every brain region, and more or less awaits its fate, which is to be broken down by monoamine oxidase, even here in the presence of a certain amount of harmine, which partially blocked the metabolism, yeah? So basically, neither citalopram, the SSRI, nor DTBZ, which is a vesicular blocker, despite having profound effects on serotonin in its metabolite, they had no effect on DMT metabolism in the rat brain. So it was a nice theory, but we can reject the hypothesis that serotonin neurons compartmentalize DMT entering from the periphery. However, I still think there's more to probe in this, and so I'm planning to go back to Denmark to, well, kill more rats, if you must know, but to get at some other things. And I'm just going to leave you with one final slide showing another very classical neurochemical tool used since the 1960s, actually, to probe the turnover and metabolism of neurotransmitters. Now, as I've told you, serotonin is metabolized by MAO to yield the metabolite hydroxyindoleacetic acid, which is a carboxylic acid. And it's not free to diffuse because it's highly charged, so it doesn't cross membranes. So any HIA, 5-HIAA, formed in the brain would stay in the brain, except for the fact that there's a transporter in the blood-brain barrier for a class of molecules that are known as acidic metabolites, like hydroxyindoleacetic acid and like the DMT metabolite. And since the mid-20th century, it was known that this transport process could be blocked by probenecid, which is itself a carboxylic acid drug, and it interacts with this transporter in the blood-brain barrier and in other organs and blocks the transport. Incidentally, probenecid was used and is still used clinically because it also blocks the transport of penicillin in the kidney. So by giving probenecid, especially in the early days when penicillin was so expensive, you could potentiate its effect by disabling the elimination. And in this classic paper by me, we measured in cerebral microdialysates the concentration of the serotonin metabolite here at baseline, and then we gave the probenecid, and as you can see, it starts to rise linearly. Why? Because it's still being formed continuously from serotonin, but it's not free to leave anymore. It's stuck in the brain, so it accumulates linearly. And so basically, this slope tells you what the rate of serotonin metabolism is, because every single molecule of serotonin eventually is converted to HIAA, and you can so extensively block the elimination process that it accumulates linearly. So this slope is basically serotonin synthesis rate. And so I'm going to do the same thing but with DMT and calculate, in effect, the kinetics of DMT turnover and to test the hypothesis that even though DMT levels are very low normally in brain, there is some production, and I expect that we will see an accumulation of the metabolite in the brain of these rats. And so that's about all I have to say, which is fortunate because the apéro calls, and I am out of time. But I have to, of course, acknowledge my co-workers in this project, and that would principally be Clemens and my host, Mikkel Palner at the University of Southern Denmark, and these people from Zurich and from Chantal Martin-Silk actually suggested the project as we fleshed out the concept together, and she's at the University of Fribourg. So I always end with this little pretentious quote from poetry by T.S. Eliot from The Wasteland, but as if a magic lantern through the nerves in patterns on a screen, would it have been worthwhile if one should say, that is not it at all, that is not what I meant at all, and PET is the magic lantern. Thank you very much for your attention. I'm not sure if you plan to take questions or...
Yes, we're planning to take questions, exactly. Right there. Let's get a microphone to you.
Oops. Sorry about that.
Hello, thank you for your talk. On the slide, not
sorry,
the last one.
Who is speaking? Oh,
Me.
there we are. Yes, hello. Hi.
Not the last slide, but the one before, that you're measuring the DMT in different brains sections. Did you also measure, after the DMT, also the serotonin in these ones? Because I mean...
That was the preceding slides. So we measured what we call the endogenous substances, serotonin and its metabolite, and the exogenous substance, DMT, and its metabolite in the same brain samples. And of course, these rats were treated with DMT in order to, otherwise there wouldn't have been anything to detect much. And they were treated with these other challenge drugs to see if we could push the metabolism one way or the other to test this hypothesis that DMT was being stored in vesicles of serotonin neurons, a hypothesis that I no longer see any support for.
Yeah, but even if DMT does not bind to the serotonin receptors, of course, like in brain areas that serotonin is very important.
Yeah, of course we thought about that as a possible explanation, but if you figure out the concentration of serotonin receptors, it's orders of magnitude lower than these chemical concentrations. Mm-hmm. So receptors are not really that abundant compared to their neurotransmitters. They occupy a privileged position, but there's about 100 times as much serotonin as there is serotonin receptors in brain. So, in effect, any binding of DMT to the receptors doesn't really show up. It's too small to measure.
You presented a lot of studies that, as I kind of thought, could lead to a hypothesis that 5-HT maybe isn't such a big part of the way DMT acts in the brain. Maybe I'm a bit... when you talked about the myo-DMT marker and the catanserin marker, and that it at the end didn't really show a correlation with DMT, 5-HT, did you at any point asked the hypothesis if it could not have an effect on 5-HT receptors?
A priori, DMT would be expected to bind to the receptors because it has a certain affinity in vitro. And for another reason, that is, there is one report from researchers in Sao Paulo where they studied the pharmacology of natural ayahuasca, the actual authentic stuff, not these chemical substitutes. And the participants were administered this blocker of serotonin, ketanserin, which is safe to give to people. It just lowers your blood pressure a bit. And they found that ketanserin did partially block the intensity of the subjective experience, but it didn't block it completely. As far as I know, I'm not an expert on the phenomenology of these drugs, but I think that ketanserin is basically known as a rescue drug for people who are having a bad experience, because with psilocybin or LSD, because it completely knocks off the drug from the receptors. But it doesn't seem to have that complete effect based on this one study from South America. So it leaves open the possibility that part of the phenomenology is related to other receptor types. But there's also methodological issues, and it gets into some complicated stuff about agonists versus antagonists, because DMT is an agonist, it activates the receptors, but the tracer ketanserin that we used in the rat study is an antagonist, so it doesn't activate the receptors. So the DMT might be binding functionally to a small subset of the serotonin receptors that are in a high affinity state and are coupled to second messenger systems. So that might be an issue. And Guidemo's Knudsen lab in Copenhagen, who was this huge operation, spies have told me, well, she told me, that they have obtained data in pigs where they used not an antagonist PET ligand, but an agonist that should be binding to the same subset of serotonin receptors that DMT is acting at. And they found that they were able to get some displacement. So I'm not saying that it doesn't act at serotonin receptors, but it acts differently than the other classical drugs. Because, as I said, with DMT, with psilocybin and with LSD, it's very easy to show that these drugs bind to serotonin receptors in vivo, but it's been more difficult to show the same phenomenon. So there's something different going on with DMT. And my initial attempt to explain that was that it was because maybe DMT was being compartmentalized and trapped in serotonin neurons, but that proved not to be the case.
Okay. So regarding the paper about the fluorodeoxyglucose study,
The which study?
Fluorodeoxyglucose study, and
yes, the Vollenweider paper.
exactly.
Yes,
So it's written psilocybin model of psychosis. So does the glucose metabolism have something to do with psychosis, or what was it relating to in the title?
It was 25 years ago, right? And I think he would probably no longer consider it a model for psychosis. That was kind of the thinking from the late 20th century. I mean, I haven't asked him, but I think he would moderate his terms now. But the idea was at that time that psychedelics were a kind of a model of psychosis, but they're not really. I mean, the whole experience is quite fundamentally different. Yeah. But that was his thinking at the time. Yeah. And excuse me, yes, that he might have expected, I suppose, that the drug, psilocybin, would alter brain glucose metabolism in some of the same areas that are implicated in psychosis. Yeah.
Okay, so I'll ask, you mentioned briefly that, you mentioned briefly that DMT really is produced in the brain, and that, first of all, that always felt like a sort of, I was never sure if that was a myth, so that would be just my first question. And since it is found in the brain, do we know anything about why it might be? I mean, we could approach this question philosophically, but maybe you have something from a biochemical standpoint to say about it.
Well, the enzymes are there for making it from tryptamine. It's a methyltransferase enzyme that was discovered in the 1950s, actually, in brain homogenates. the discoverer has slipped my mind, but he got the Nobel Prize actually, not for that discovery, but for basic neurochemistry. So the enzymes are there. The concentration of DMT is possible to measure it. In my rat studies so far, it was there sometimes, it was on the edge of what was detectable. So the levels are low. And the reason the levels are low is because it's being formed, I'm convinced of that, but as soon as it's formed, it's broken down by MAO. So I think that DMT formed in the brain might assume a physiological importance in the condition of MAO blockade. And so I'm going to do further rat studies to try to get at that. But I think the amount of DMT that's present, unless there's something that we really don't have an understanding of, which is always possible, I don't see that those low levels could be doing anything. Philosophically, everything doesn't have to have a purpose. You know, there can be there can be enzymes that are just there for some other reason, and they happen to produce this result of producing DMT levels. Because the enzyme that does it is also N-methylating a lot of other substrates. So the DMT might be a happy accident, let's say. But I'll take the opportunity to dispel the idea that was promoted by some people that the pineal gland produces DMT and that it causes near-death experiences. You'd have to, there's nowhere near enough there. It is at moderately high concentrations in the pineal gland, much higher than in the brain itself. But I still don't think that the amounts present in the pineal gland, maybe a milligram in total or less, could be really functionally significant, unless it has some action that we don't know anything about yet. That could be.
You mentioned that it's kind of surprising that those Mesoamerican cultures figured the two plants which they needed to mix to get ayahuasca, the one with the DMT and the one with which inhibits the decay of DMT. Now, wouldn't you expect that they, on some point, would just by coincidence figure that out if so many people live for such a long time, eating whatever grows in the forest, that they would mix by coincidence this kind of two plants? Or why do you think it is surprising?
Well, there's probably at least 100,000 species of plants in the Amazon they had to find. But it's not that simple, because it's not just binary. There are alternative ayahuascas that have different plant constituents. Like, for example, the mimosa contains plant from Central America, contains DMT. That's actually where the University of Zurich gets their DMT, is they extract it not from the ayahuasca plant, but from another plant. So that increases the odds, the fact that these compounds are not uniquely present in just one species. But, I mean, in their narration, the plants told them. But I'd like to believe that. But I think that it seems to me a remarkable thing. I mean, they knew their jungle. This was their livelihood. So they knew what plants would be safe to eat and what plants were medicinal. Probably, I'm just speculating, but maybe these plants had another function. Maybe they were antihelmetic. So, a lot of tropical plants have these kinds of anti-parasite effects. And so there was probably some reason why they converged on these two plants. But I don't know if it's possible to reconstruct thousands of years after the fact. And I think the archaeological evidence goes back at least 1,000 years in the cultural evidence for the cultural use of these mixtures. But there's way more to it, because the composition of natural ayahuasca mixtures is highly variable, and it depends on the shaman and the region of Amazonia. And some people even say that DMT, this is an extreme position, I have no opinion on that, but some say that DMT is not even an essential ingredient. I don't know what to say about that. But so it's not off-the-shelf chemicals. It's, they hit upon these customs based on their ancient traditions and experience. But, well, as far as I know, the coca leaf chewing is typically done by chewing along with burnt seashells, right? Calcium carbonate. Yeah, and that's the pharmacokinetics or dynamics is that if you basify the mixture, the free base is more readily absorbed across mucous membranes. It's just an example, but how did they hit on that? How did they decide that cocoa leaves would be more potent stimulant if they were chewed along with calcium carbonate? I don't know. They only had to, I guess, they only had to do it once and notice that it worked.
Okay. Due to time reasons, we're 15 minutes over time. We'll wrap up here. Thanks, Paul, so much for coming, for giving this beautiful talk. Thanks, Jo, for joining us. If Paul is around, then we can all ask him, bombard him with our questions at the apéro.